Hey, it's Mr. YS, and this is a clip explaining the question to do with Meselson and Stahl. So what they're asking you is, what is the density of the DNA at generation 1? And they give you some information. And they tell you that the density at the start was 1.724. And this was for cells that had been reproducing in the heavy N15 environment. So if we imagine that this we label as red, so heavy N15, that means both their strands would be the heavy N15 DNA. And so we know that that density is 1.724. The other bit of information they give you is that after 4 generations, they tell you that this dark band, the density of that is 1.710. So if the density of this dark band over here is 1.710, well, this band, if we look over here, if we kind of turn it to the side to imagine our test tubes, where we would, or our centrifuge tubes. If we imagine this versus our initial banding experiment, what we had in the initial experiment was that the DNA, which was 2 strands of the heavy N15, would move down the centrifuge tube because it's more dense. So the lighter strands would be further above. And so what they found was, after copying it in an environment with only N14, so I'm going to do that as this light blue, if we only have nucleotides with light blue, then here's our original heavy N15. If we open up those strands, then the only thing that we can put in is the new lighter N14. And so after 1 generation, they found a band, all the DNA is 1 light N14 and 1 heavy, so that's the heavy N15. So if you are half light, half heavy in terms of the N14, this is an N14, N15, so this is a hybrid. This is the intermediate density. Okay, so what we should see after generation 1 is that the band there is showing us the density when we've got half light, half heavy. The band here was showing us all heavy, and now this light band down there, well, that's if we've copied again. So if we copy this strand, if we open it up and copy this strand, so the red will go with the new light blue, we would still get a faint band showing our hybrid. But the one over here, the blue strand, is going to be copied with a new light blue strand. And so we're going to get this double N14. And so what we're seeing here, this darker band, is just 2 of those lighter N14. So they've told you the density of that is equal to 1.710. And so we're trying to find the point halfway between, when it's half heavy and half light. So we need to get a density halfway in between those points. And if we take a density that's halfway in between, one way to do that is to just add them together and divide by 2. So 1.724 add 1.710 divided by 2, and we get 1.717. The other way is to think about, well, what's the difference between these? The difference between them is 0.14, so that's the total difference, and half the difference would be 0.07. And then we can add that on or take it away from the top one. And if we do that, we get the same answer. So quite a difficult question, because it's, you ignore all this, these in-between generations. You're just trying to remember that in the Meselson and Stahl, in the first generation, so after copying once, we have hybrid, and that's going to be halfway between the density of either the fully heavy or the fully light. In the next part, they ask you to explain the banding pattern at generation 3. And so the banding pattern, whenever we're talking about bands, and we get these banding patterns also in genetic questions where they run things out, we can see that the darker band, it equals more of whatever that thing is. So in the case of this is more DNA. There's more DNA in that darker band, and this lighter band means there's less. So we're trying to explain, why is there a darker band? Why is there more DNA here, further up the tube, and less DNA there, as compared to at generation 0, where there was only one dark band and it was further down. So towards the bottom is more dense. So we've got more DNA in the lighter part, the less dense DNA. And this less dense DNA is the 2 strands of N14. So we're trying to explain, why did it go from having a darker band here, which was just all the N15 heavier, to, and you can see there's a kind of a range we get. If we look to where this band was there, it's kind of spread out, but we've still got the intermediate. We've still got a bit of a band of the intermediate as well, which is this, which is 1 strand each. So we're trying to explain that. Why has it gone from that to now having most of the DNA being this, the lighter? And so this is where we talk about the fact that DNA replication is semi-conservative. If we take the 2 strands, which are red, if we open them up, we use the heavy strand as a template for the synthesis. And if we're in an N14 medium, the only nucleotides that we can put in are the lighter N14. And so each time, if we imagine what's happening here, we are halving the density, or halving the number of heavy strands here in terms of the total. So they started out here, we've got 2 out of 2 are heavy. 2 divided by 2, well that's all of them. Now here, if we have a look, we've got 2 out of 4. If we copied again, we'd open up those red strands, we'd add in the blue, but then these blue strands in the middle, they would be paired with a new blue. And so now we're going to have, in terms of the heavy, we're going to have 2 out of 8. So we can see each time we copy, we're going to half the number of the heavier strands. And so progressively we're getting more and more of the lighter strands, and this is what explains this band. So each time we're getting more of the light N14 strand.